Cette conférence va maintenant être enregistrée.
Sorry, I just forget to record the conference. Please continue, sorry.
No, thank you so much. So, and you can see the Réseau, Francophone, Métabolomique, and Fluxomics. Okay, so there is very across the globe, different groups that apply metabolomics in a different field and translation. Just to make sure that we are speaking the same language, we understand metabolomics as a omic science that intends to understand the chemical space of any biological system. And this chemical space or molecules can be a metabolome internally, endogenous, or exposome, or metabolites that also are excreted in the media. And this chemical space, it has a very interaction with other biological layers, be on a genome level, transcriptome, and proteome. And it's a back and forth interaction. And measuring this chemical space, which gives us what some call the biochemical phenotype of any biological system, we are able to understand what is actually happening at that specific time under any specified physiological conditions. This diagram just to show how complex these interactions are from the genome level to metabolome, and how they really switch on each other and influence each other. So what makes metabolomics, okay, there's someone talking in the background somewhere. It's a multidisciplinary science using different domains, computer science, biology, chemistry, statistics, and a successful metabolomic study, as you all would know, it really implies a team efforts from different specializations. It has grown in the last 2 decades. We really went look into number of publications that put forward very so rapidly. And however, despite that growth and momentum of the metabolomics of the science, we still really have challenges. The chemical space is so diverse and so big, and so far we are able to understand or to annotate or to know a very small fraction, be in a different biological domain, E. coli, human, or plants. However, with the efforts, community efforts from different domains, the use of computational strategies is really providing opportunities to be able to tap into this chemical space. Okay, number background noise. Okay, that's fine. So as I was saying, the emerging of computational strategies is helping to really be able to tap into the chemical space to elucidate or annotate the dark matter of that chemical space. In that way, also improving our biological understanding of any biological system that we investigate. And just here. Okay. Okay. So just look here in the last decade or so, we can see how if you just looking for computational metabolomics expression in a publication, it's really a huge exponential growth. So in our research, we have these 4 focus area. Contribution to the methodology development can be in data mining strategies, be computational tools, or investigating machine learning or deep learning algorithms and analyzing statistically the metabolomics data, natural product research, and plant biostimulant interaction, and microbial metabolomics. in this presentation, I will just simply share with you these 2 points. What selected studies that we have done, where we see the application of computational metabolomics to be able to derive biological insight into that helps to answer specific questions. Plant biostimulant, how we look at the 2 studies, use 2 studies to showcase such kind of use of computational metabolomics. We, in one study where we look at the microbiochemical space, focusing on rhizobacteria, plant growth promoting rhizobacteria, and also looking at microbial biostimulant and crop-plant interactions that we have done. Just a bit of background when it comes to biostimulant. You would agree with me. Agriculture is one of the cornerstones for our human survival, for food security, economy, and environmental sustainability. However, there is, or it has been, or there still are some challenges in agriculture due to different reasons: climate change, the water scarcity, soil degradation, rising input cost, etc. And the FAO has predicted that this food insecurity has risen 4% just in a short period of 3 years. So there is a need, this is a challenge that the humanity faces, and different stakeholders come on board to find ways that we can tackle provide some solutions. So some of the solution could be around finding sustainable and precision agriculture methods. And in this case, biostimulant comes handy as an option, but we're still not understanding the mode of action of these microbial or non-microbial biostimulant products. And this using the current technology we have, the research we have, the data analytics. In this case, what we generate from metabolomics studies, we can provide some solution towards crop management, resource usage, and also decision making. In metabolomics, as you all know, as I said in the previous slides, where I showed that studying this chemical space gives us really a snapshot in what is actually happening. Such information is really useful in a way that we can be able to come up with data-driven approach to design either biostimulant formulation for better application, or also program to apply this biostimulant. So in this presentation, I will just share about the microbial biostimulant. These biostimulants are normally divided into 2 categories: non-microbial, that would be an example of humic substances, or microbial, it can be bacterial-based, like rhizobacteria or fungi. So in this example is microbial biostimulant that we're investigating. And firstly, here we want to look at what are the chemistry of this bacteria. We looked at bacillus strains, liquid culture experimentally, and we looked at intracellular metabolites from in the bacteria, and also the metabolites that are excreted by this bacteria. Why would one want to look into those chemical entities? These metabolites, or these chemical compounds, are the ones that the bacteria use to communicate with another bacteria in the surroundings or with the plant. So looking at these bacillus strains, did just a broad study, liquid culture, the samples we harvested at specific time points, extracted, analyzed on the LCMS system, the spectral data generated. We also apply computational tool strategies. In this case, molecular networking to annotate those metabolites. And as you can see here, there's really this bacteria do contain or produce a wide range of chemicals or metabolites of different classes, lipid and lipid-like metabolites, organic acids, etc. Our interest in this case would be lipopeptides, which are have been reported to have different properties in the interaction of the microbe and plant, and also in the microbe-microbe interaction in the rhizosphere. And this diagram, just to demonstrate if one can try to understand schematically, the root zone. We have those bacteria. They can be endophytes or exophytes, and these bacteria do secrete metabolites, and also the roots do also secrete metabolites, which leads to this chemical communication between the bacteria and the plant. So understanding this chemical communication, it helps to derive the mode of action of this biostimulant, microbial-based biostimulant, and the benefits of this interaction with the microbe and the plant of different levels, can be growth promotion or defense priming. Defense priming understood as if one was to make an analogy to the human, would be like vaccination, biocontrol, nutrition or nitrogen fixation, etc. So the benefits of this interaction of the microbes at the root level is quite a lot. So then we look also not only to understand the chemistry of the microbial system, but also to look at above ground, the interaction between the plant and the biostimulant. We also focus only on the microbial, the non-microbial studies. It's for maybe another time. So we use this so one big study, but that was divided in 3 small studies, where we looked at the plant leaves responses to a treatment with a microbial consortium. Also, we looked also at the sap in that same system, maize plant, and also we targeted a specific class of metabolites, and also we applied epigenetic studies to also look at the gene level expression, what happened. This was a greenhouse study, and these are a number of different classes of metabolites that we annotated, and look at the pathway, biological pathways that are involved in the changes, the metabolomic reprogramming induced by the treatment in this case being microbial biostimulant applied to the maize plant. And we see indeed different pathways, some involved in the enhanced defenses, others involved in growth promotion events. So we came up with this model that explains I just here say PGPR, but it's a microbial formulation that has been put on the market, but to understand the mode of actions. So this is a model that resulted from these studies that explains at the molecular level what happened in that interaction of the plant in this PGPR-based formulation. So we see changes at the genetic level, global methylation, but also with the specific genes changing. And also in the sap, we see change in the different classes of metabolites that are carried from one part of the plant to the other. And also in the leaves, also we see a number of metabolites that have been changed in the different pathways. And these pathways are involved in different physiological events. And some of these antioxidant defense, enhanced energy production, controlled respiration, increased leaf, shoot, and biomass. So these studies have provided insight on the mode of action of this specific microbial biostimulant, which was bacillus-based, and providing a model that explains how this product works at the molecular level. And in that way, the company that used this formulation is able to design programs that can be used for a precision application of this formulation. So we also, as I said before, when I was sharing with you about the chemistry of the microbes of this microbial biostimulant, we saw different classes of metabolites that are internally or excreted by the microbes, and this points to a chemical communication or elements involved in the chemical communication between the plant and the microbe. So we did another study where we wanted to investigate the below ground chemical communication between that specific microbial biostimulant and the maize plant. And we, on the same protocol, we get the samples, analyze on LCMS system, and analyze the data using computational strategies. Just an example here to a molecular networking that shows a number of classes of metabolites that we were able to annotate from these interactions. And here you can see the consortium of that bacillus strains and individual strains, but some of them are part of this consortium. The control is were not treated, and we can see the changes, the distribution of those metabolites depending on different conditions that were applied. So what we could understand from the results that we saw is the changes in the chemistry of the rhizosphere, meaning also surely the plant also does excrete some exudates in the compounds that act upon on the microbes, and that chemical communication leads to the results we see. And this class of compound, for instance, they are involved in nutrients acquisition. And here also we can see different events, communication via the excretion of secondary metabolite, the disruption of the plasma membrane, and also the contacts to cell-cell communications. So in a larger context, such study provides understanding of the dynamic chemical communication between rhizometabolites, can be those from the microbes or those from the plants. And also we are able to decode this chemical language that can help to understand the mode of action of this biostimulant. And with that, we are able to provide information that can help to design novel bacillus-based formulations or products that can be put on the market for sustainable and precision agriculture. So this study of biostimulant, it's really our studies just to contribute to a global ongoing efforts in trying to understand these formulations that have been proposed as an alternative for sustainable agriculture, and in a different from different countries or different globe. There are different studies that are being done. So some of these studies were funded by Innovate UK and in a collaboration with the University from Edinburgh and the ACGT and the company's Omnia. So that's just the studies that demonstrate the way we use computational metabolomics approach to investigate, to study biostimulant, either from understanding the biostimulant composition or the chemistry or the biochemistry of the biostimulant formulation, or also to look at the plant biostimulant interactions to understand how the plant responds to this treatment, and also to be able to decode what are the possible chemical communication or molecular communications that take place that drives the physiological changes that we see or we want to see. The second part of the research focus area, as I said, is a plant-derived natural product that we do. And I'll start by sharing this recently published protocol in a book chapter that provides step-by-step the use of computational metabolomic strategies to study plant-specialized metabolites. And this was an effort involving the research group of Thomas Pluska, he's in the Czech Republic, and Justin van der Hoeft in Netherlands. So please have a look. It's already we tried to really make it easy for most of the community are not computational scientists, but the protocol provides step-by-step ways that one can apply computational metabolomic strategies to study specialized metabolites. Also, another method-like type of work where we use mostly mass spectrometry as an analytical platform. And as most of you may know, there are different methods that we acquire spectra data. And in this case, specifically for fragmentation data, because fragmentation data gives us structural information. And at the end of the day, that's what we need, any pieces of information that can help us to annotate the chemical signals that we detect. So the two strategies that are normally used is data-independent acquisition and also data-dependent acquisition, DDA. So data-dependent acquisition has its limitations, and some of the limitations is that you are limited in the coverage of the signals that you can detect, because indeed it depends on the parameters that you would have set. So in this study, this model that we propose is, okay, can we use or combine different mass spectrometry parameters to indeed increase the coverage that one can obtain using DDA method for structural information acquisition. So please have a look, and it helps. Natural products are from different sources, as you may know, from plants, microbes, etc. And they have been really traditionally from years back been involved in a medicinal application. The traditional medicine from different societies indeed has played a role to keep human being alive and treat different diseases, etc. And it has different ways from different cultures, different societies, and the way people really explore this natural product from different sources. In our research group, we focus so far on plant-derived natural product, and examples of this medicine from the plant are many. Morphine, for instance, and that has been isolated from plant. And the natural product research at some point got a little bit slowed down because this was due to possibly the technology of the then that led to more re-isolation of the same kind of bioactive compounds. And now, but however, recently we see a momentum, a growth again, or a visit of this natural product research. And this can be attributed in one way to advanced technologies that we have. We live in a digital age. We are so privileged to have different tools now that can be used to indeed study or get quite a lot of information. So we participate or try to contribute to this effort, global effort, to look at plants that have health benefits properties and be able to try to understand the chemical space of these medicinal plants with the aim to contribute towards drug discovery. So in this presentation, this part of the presentation, I will only talk about these two plants found in South Africa, Mamodica plants and the Helichrysum. And the Akhona will talk about cannabis research that we have done. So the Mamodica plants, this is a publication by this PhD student. The four Mamodica plants that we looked at, which are found in a Venda region. It's in the north part of South Africa. And they have some of the claims from this plant is this anti-malaria properties, anti-inflammatory, and anti-diabetic. I'll put this in blue because that's what has been our focus for this study. So we did characterize the chemistry of this plant just a little bit. This is not really well characterized. So we provided for the first time a metabolomic map of this plant that can provide insight into the chemistry of the plant. So applying, again, computational metabolomic strategies, we were able to map or to annotate different classes of metabolite from this plant. And also we can see on this diagram, the distribution of these chemistries is really from different plant species. It's very differential distributed. Again, these are a number of the metabolites and classes that we were able to annotate. And also bioactivities, we focused, we did the in silico prediction, we use a computational method. This network pharmacology and the molecular docking to predict what could be the interactions and the properties related to the bioactivity here. The focus has been anti-diabetic potential of the plant. So the student will be submitting her thesis soon and possibly publications also to share what we have found. Another study is this Helichrysum plant that are found in South Africa also. So we looked at these three species from this plant, and with the same applied computational method to annotate, to characterize the metabolome of the plant, and also to predict the pharmaceutical potential of the plant. This from this less studied species, Helichrysum splendidum, we could see different superclasses, this molecular network, MOLNET enhancer, where we see lipid-like molecules, phenylpropanoid and polyketides. We zoomed into these two superclasses because that's where we want us to narrow the study in terms of the activities of the plant. But as you can see, the chemical space of this plant is quite vast and diverse in terms of the number of different classes of metabolites. So one would assume the activities of these bioactivities from this chemistry or phytochemistry of the plant is possibly quite a lot. So we focus on this because here the intention also was to focus on the anti-cancer properties of the plant. And we looked simply only at those two classes of compounds. We also explore, as you may know and understand, one of the challenge or what makes metabolomics a little bit challenging, the chemistry, these small compounds and metabolites, they have a diverse chemistry and diverse physical properties. This affects the way we can be able to understand this chemistry. It can be at different stages of the metabolomics workflow. It can be at the extraction level. It can be at the analytical level, the instrumentation, where you analyze the samples, and also at the data analysis level. For extraction, you would agree with me, depending on the extraction method you apply, you are biased to the type of chemistry or the metabolite you are going to extract from your biological system. Again, on the analytical level, depending on the, for instance, on chromatographic space you use, is reverse helix or non or direct infusion, that also will indeed affect the chemistry that you will be able to annotate, etc. Again, also, as I referred to the mass spectrometry DDA parameters studies that we showed, depending on the way you set your mass spectrometry acquisition methods or aspects, it will also define what metabolites at the end will be able to detect. So all those aspects do really indeed make pose a challenge in the way we try to annotate this metabolome on any biological system. So in this case also we want to say, okay, there's these helichrysum plants that we are investigating, we're looking at, etc. What ways that we can indeed increase the coverage? Did use different extraction solvents to be able to tell to see what increase the coverage. For instance, as you can see, this class of compounds will only be able to extract them using hexane. So if one would have only done methanol extraction, methanol-based extraction, there is indeed, for instance, one would miss out such class of compounds that are produced, that are found in that plant. So also the same, if you look at the distribution of those class of compounds, the three plants, very indeed different. These were extracted using hexane, non-polar solvents, and we see most of them are found in the helichrysum talicum, and these ones which were extracted using ethyl acetate, most of them found in the helichrysum splendidum. So this does demonstrate ways that one can really increase the coverage of the metabolome of interest, but also this depends on the biological questions that one intends to answer. So we did, I didn't show it here, biological assays for anti-cancer properties of the plant. In this case, we just looked at the crude extract from those different solvents and tried to see, looking at different cancer cells. This was just in vitro cancer cells. We did see some activities and also complemented also with this molecular docking, where we looked at the different classes of metabolites and the interaction with the specific proteins involved in different physiological events. From here onward, what one would need to do would be proper fractionations, target to the specific classes of compounds to narrow to a specific a bioactive compound of interest and downstream towards drug development. So however, information like this will publish to share with the community the insight we found from such study. So for the first time, we're able to show that there's very helichrysum plants have quite a lot of wide phytochemistry beyond phenolics, because previous studies, most of the studies have been focused on phenolics, and this understandably so, depending on different extraction methods used, different analytical system used, etc. So we're able to see, at least with the use of computational metabolomic strategies, a wide range of the metabolome of this plant, helichrysum, and also provided methods that can be used to study such plants and even in other plants other than helichrysum, and also contributing to a mass spectra database, because the spectra data that we generate, we do submit them to repositories that other people also can use, or it can increase spectral libraries that are available, and that would make it easier for annotation of metabolites. We always try to put in context what we do, what it means, and why. Understanding the chemistry of erythrithium contributes towards the good health and well-being, and also partnership for the goals, collaborative efforts. The work we do, I will always collaborate with different research groups across the globe. All of this that I've presented, it's not done by me directly only. It's really team efforts. It's a really wonderful team that I have, that I work with. I'm so grateful. So these are the PhD candidates and the masters. Some have finished. And the photos are uploading, they're coming soon. So I would like to end by acknowledging these people from University of Johannesburg who really assist in different ways and support, and also Omnia team. Omnia is a company that I work with in terms of plant and biostimulants. And for collaboration here in South Africa and internationally, I would like to acknowledge the following. Prof. Madara, who is at University of Venda, and he really collaborates a lot on natural product research, and also he really has expertise in mass spectrometry, and also from overseas, Justin, Carl, and Peter Dobson, that really we work a lot on computational aspects. We'd like also to acknowledge the funders, the sponsors. So beyond science, we do have sometimes some fun and also share some meal, lunch. We have a Twitter account. Please follow us and see what we do in our daily life. And the website is uploading also. Thank you so much again. And if there's any question, I will be happy to answer.
Thank you very much, Fidele. So any question from the audience? Come on, don't be shy. I have one to wait for the other ones. So you mentioned a commercialized biostimulant product, and I wonder if you have some feedback from the stakeholders after they use this in the field.
Yes, that company Omnia, they do follow up. First, I think the way they design, I may not fully understand the whole process. Once they put the product on the market, first they have a number of farmers that they target, they sell the product and they get feedback from them, and then they refine the marketing strategies, etc. So one of the studies that extension to the greenhouse studies, we did do a field trial studies, and with the same product that has been put on the market. And we looked also then at a different growth stage of the plant, from planting to harvesting, the yield. And yes, the results that we saw on the field, it's very positive, and increased in terms of the increased yield, but also the nutritional values. We measured protein content in the corn, etc., vitamins in the corn, and we could see that the application of that microbial biostimulant does really have impact on those parameters. So the company does use all those information to do the marketing strategies, etc. As far as I have understood, the product is also on the market in Australia, in Europe. So it's something positive, yeah.
Nice, thank you. Any further questions? You can ask in French.
Yeah, you can ask in French. Yeah, I may try to understand. I do understand French, but my speaking is not that good yet.
I have one, maybe just you talk about the impact of extraction methods that you can use. So what is for you the best strategy for you to have access to the most exhaustive list of metabolites?
Okay, so as I said, that's a tricky part. It depends. Also, it depends firstly on the initial biological question that you want to answer. Let's say, for instance, if I'm in natural product research and for the first time I see this plant, there's not much information in the literature about the plant, and maybe with the little knowledge, I would suggest, for instance, to use methanol extraction. It's generic. It really covers a wide range of chemistries, polar, non-polar, but to a certain extent, and that can give you just a preview of what the chemistry of the plant looks like. However, if I have additional information, for instance, I know in traditional medicine, this plant is used by boiling the leaves, then that gives me an indication of what type of chemistry that is more related to the activities claimed to the plant. Then I can then mimic or design an extraction method that would focus on that class of compound. But if I'm just doing an exercise to come up with a comprehensive metabolomic map or atlas of the plant, with time you can really explore different polarities in terms of organic solvents and increase the coverage of the metabolome that you extract. So the other thing, okay, with this computational method, advantage we have is that the way this molecular networking works, as you know, of course, it to link similar spectra. So if I can be able in one node annotate what that spectra is, it means the other connected nodes are of the same class of compound. So that at least already gives me one step in advance or ahead to annotate or to elucidate that dark matter. So even though I really don't know in the library what that, but at least I would tell this is part of this class of metabolite. So for extraction, I will start with a general extraction method that I'm familiar with. Methanol is one of them, called methanol. It really extracts a wide range of compounds, and from there, using additional information that I can gather, I can design another extraction method. Also, if I know the biology of the plant or what type of the chemistry that is found in the plants, you can really design another specific extraction method. For instance, if you look into glucosinolates, the hot methanol extraction is good to extract most of them, but surely you miss out other classes of compounds in the plants. So it's really trade-off kind of approach, I think. Yeah.
Okay.
Thank you.
Thanks a lot.
Thanks a lot.
All right, so let's move to the next speaker.
Thank you so much.
Thank you, Fidele, again.
So
And we stay in your group anyway.
Yes, okay, thank you.
So now we welcome Akhona MYOLI, which is a PhD student at the University of Johannesburg under the supervision of Fidele. She will talk about computational metabolomics in natural product research. So the floor is yours. I think you can share your screen.
Is it sharing now?
Yeah, it's okay.
Yes.
You can just put full screen.
Hello?
Yeah, perfect.
Okay, so I'm audible. Yeah, thank
Yes.
you. Okay, good afternoon, everyone. As introduced, my name is Akhona MYOLI, and today I'll be presenting on our study, which is titled Charting the Cannabis Plant Chemical Space with Computational Metabolomics. This is the outline of my presentation. The aim of the study was to simply apply metabolomics approaches to characterize the chemistries and underlying biochemistry of different cannabis cultivars that are used for medicinal purposes. To achieve this aim, the metabolic landscape of the different cultivars was assessed using LC-MS-MS analysis workflow, and then this was then followed by the structural elucidation of metabolite features that were present within these cultivars. And then lastly, in silico molecular docking studies were done in order to predict the biological activities of these plants, and this was done in conjunction with biological assay studies. Now, this project, at its core, it was based on plant natural product research, with the plant of interest being cannabis. So why cannabis? Well, cannabis is one of the plant genera that has gained attention for its bioactive natural products and its use as a medicinal plant. The extensive use of cannabis dates back to 5,000 years ago, where it was used for recreational uses, medicinal uses, and textile uses. With regards to its medicinal uses, cannabis is known to alleviate symptoms within individuals that are suffering from cancer or HIV, and it's also known to alleviate some neurological symptoms as well. Now, all these therapeutic properties, or all these observed therapeutic properties, they tend to be attributed to a class of compounds called cannabinoids, and in particular, compounds such as CBD and THC. And evidently enough, numerous drugs have been developed from these compounds. So given these examples and some of the drugs that have been developed from cannabinoids, one can see that past research and some of the current research that are ongoing today mainly focus on cannabinoids. However, the phytochemistry or the metabolome of cannabis is not fully understood, nor has it been fully explored. And if we're going to consider cannabis as a medicinal plant, it is very important to note that there are other chemical classes within this plant beyond cannabinoids. So therefore, the chemical exploration of cannabis remains important, because the information that we can generate from such studies can help us to better understand the biological properties of these plants, and it can also lead to the discovery of novel compounds. Keeping that in mind, metabolomics has been suggested for the chemical exploration of cannabis, coining the term cannabolomics. And cannabolomics as a field, it aims to explore the chemistries of cannabis varieties with the ultimate goal of revealing new drug candidates within these plants. And additionally, metabolomics has been suggested because it offers us a wide range of advantages with the use of its highly sensitive analytical tools as well as robust computational tools. So in the study, we took full advantage of the metabolomics computational tools such as feature-based molecular networking and MS2LDA. Feature-based molecular networking, it is a computational tool that works on the basis that compounds that are similar in terms of their structure will fragment to produce similar fragmentation patterns. And then based on those MS fragmentation patterns, the compounds are then grouped into one molecular family. And additionally, feature-based molecular networking also enables the automated annotation or identification of compounds within your samples. So it also then allows you to visualize the sample space that you are working with without knowing the chemical composition of your samples. And then secondly, we made use of MS2LDA, and MS2LDA is a substructure discovery tool, which is unsupervised. And how it works is that it will annotate the building blocks or the substructures of the compounds, therefore propagating the identity of those compounds. So both feature-based molecular networking and MS2LDA, they actually assist with the dereplication of compounds. And we know that in natural product research, one of the biggest challenges that we face is the lack of dereplication of compounds, which often then leads to the re-isolation of known chemical entities instead of putting more focus in discovering the novel compounds within those biological systems. Now, both these tools are free to use, and they are made available within the web-based ecosystem, which is called GNPS. So because of time, I'll skip through the methodology section and go straight into the results, which is the most important part. So as mentioned earlier on, in this study, we were looking at different cannabis cultivars. So we namely looked at the leaves and flowers of Amnesia haze, as well as the leaves and flowers of Royal Dutch cheese. And from the LCMS data that we had acquired, we saw that this data was showing us cultivar to cultivar metabolite differences, as well as plant tissue to plant tissue metabolite differences. So now, to better understand these differences, we had to perform metabolite annotation. And as mentioned, we made full use of computational tools such as feature-based molecular networking. So using feature-based molecular networking, we were able to identify the cannabinoid cluster within these cultivars. We were able to identify this cluster as being cannabinoids because one of the nodes or metabolites in this cluster, it was automatically matched through GNPS spectral library matching as being CBD. And then this then propagated the identity of other cannabinoids in this cluster, example being your CBDA as well as CBDVA. Then additionally, we were also able to identify the flavonoid cluster. And as you can see, all the nodes that are circled red, they were flavonoid compounds that were automatically matched through GNPS spectral library matching. And then this then propagated the identity of unknown metabolites, such as your canaflavin B, which is actually a unique flavonoid that is found within cannabis plants. Then when it comes to the use of MS12DA, when focusing on the flavonoid cluster using MS12DA, we were able to also enhance the annotation process, right? So when looking at this flavonoid cluster, we saw that there was a metabolite, which is in the middle here, and this metabolite was originally unknown. And this metabolite was also then connected to two other metabolites that we had previously annotated using feature-based molecular networking. So with MS12DA, we were able to see the substructures of these compounds, and in particular the substructure that was common between the two known metabolites and the metabolite that is unknown in the middle. So with MS12A, we were able to see that this unknown metabolite shares a common substructure, which is in pink, with the two other known metabolites, and that common substructure was luteolin related. So this then propagated the identity of this metabolite to be luteolin glucoside arabinoside. So this is an example of how MS12DA can actually aid with the dereplication of compounds by enhancing the annotation process. And then additionally, MS12DA also gave us confidence in the annotations that we had made, right? So if you look at this cannabinoid cluster, the substructures that were elucidated in this cluster, they correlated to the structures that we annotated, right? So that then also gave us the confidence to say that the annotations or the putative annotations that we were making were correct. Right. So all in all, the use of feature-based molecular networking and MS12DA, they helped with the dereplication of compounds, and they also helped us to identify the different chemical classes that were present within the plant tissues of these cultivars. So essentially, we were able to use these metabolomics tools to differentiate the two cultivars based on their chemical content. So meaning that we're able to classify these two cultivars into chemovars, right? And the next question would be then, what is the significance of this chemovar information and how can we apply it in the real world? And the answer to this is that this type of chemovar information, it can help us to narrow down specific cannabis cultivars for specific medicinal applications. So what do I mean by this? So again, when we zoom into the chemical profiles that we were able to elucidate, we saw that the leaves of these cultivars was producing more of your non-active cannabinoids or acidic cannabinoids. And then when looking into the flower content, we saw that the flowers were producing more of your active cannabinoids, such as your CBD and CBN. So if an individual is looking for your cannabinoid biological activities, for instance, if you are looking for the CBD biological activities, where CBD is known to have anti-inflammatory properties, then you are better off using the flowers of these cultivars instead of using the leaves. But you are better off using the flowers of Royal Dutch cheese, because we can see that in Royal Dutch cheese, CBD was produced in higher levels compared to the Amnesia haze cultivar. Then again, when looking at the flavonoid content, we saw that the leaves of the cultivars were producing more of your flavonoids compared to the flowers. And again, if an individual was looking for flavonoid biological activities, such as antioxidant activities, then they are better off using the leaves instead of using the flowers, right? So now, given these chemical profiles that we were able to elucidate, we then went on further to perform in silico molecular docking studies in order to predict the biological activities of these cultivars. And from these in silico studies, we saw that when we were testing whether these cultivars could have anti-cancer properties, we saw that the cannabinoids had the most significant binding scores to their cancer targets, but they were also closely followed by flavonoids, such as your canaflavin A, which had a very significant docking score of minus 9.3, right? So these in silico tools, this in silico study, it also then alluded to the notion that these cultivars do indeed have some type of anti-cancer activity. So to further support these in silico studies, we then performed biological assay studies. And for these biological assay studies, such as the caspase activity, we chose to continue with the flowers of these cultivars because, number one, individuals that take cannabis as medicine, they usually use the flowers instead of the leaves. And also, most of the clinical research that is out there will mainly focus on the flowers instead of the leaves. So with this caspase activity assay, we then took the flower extracts of Royal Dutch cheese and Amnesia haze, and we treated a cancerous cell line, which was the PACA2 cells, with these flower extracts. And what we saw was that in these cancerous cells, there was an increase in caspase activity when they were treated with the flower extracts of Royal Dutch cheese and Amnesia haze when compared to the untreated cancerous cells. So an increase in caspase activity is indicative that cell death was occurring within those cancer cell lines, and that cell death could be due to apoptotic events, necroptotic events, or autophagic events. So this type of study then also just gave us the idea that these two cultivars do have some potential when it comes to anti-cancer properties, and it also qualified them for further studies such as fractionation to actually identify which compounds are responsible for what we are seeing in terms of the biological activities. And it also qualified them for further studies with regards to testing their anti-cancer activities. Which then brings me to the significance of the study. So the significance of the study is that we were able to utilize Métabolomics computational tools and strategies to classify the cultivars into chemovars, and also the chemical profiles that we were able to elucidate from these cultivars gave us more insight into the potential medicinal properties of these plants. And lastly, the type of work that was done here, it also contributes to cannabis research and development, which is currently on the rise. And one way in which we've contributed to this movement was through publication. So this work is also published. So I would highly recommend that you go and read this paper for, more information. And yeah, so that is the end of my presentation. These are my acknowledgements. And thank you so much for listening and for inviting us to the webinar.
Thank you.
So thanks a lot for your presentation. So is there any question? If you want to, don't use the chat and you want to open your microphone and ask, you can. No, not for now. So I have one. So what is your plan for this study? What do you want to do next to continue this work?
This work is I think my next plan would be to actually focus on studies such as fractionation, try to narrow down as to what could be the compounds that are responsible for the predicted anti-cancer activities and what we had seen in the biological assays that we're doing, because we were using crude extracts. So now you don't know what is actually contributing to the results that you're seeing. So doing fractionation studies, I think, would be the next valid way in which we can explore these plants.
Okay, thanks.
All right, so our next speaker is Salek Ahmed Sajid. He's a third-year PhD student at the IPS2 Institute in Université Paris-Saclay. So he's working on the role of biogenetic organelles, plastid and mitochondria, on scotomorphogenesis in the model plant Arabidopsis. So Salek, please, the floor is yours.
Yeah, thank you, Pierre. Can you hear me?
Yeah.
Yeah.
Okay. So let's share my screen. Yeah. And can you see my screen? Yep.
Yeah.
Okay. Great. Thank you, everyone, to be here, and I'm very happy to present the results, some of the results of my PhD. So my PhD is to identify the role of bioenergetic organelles, the plastid and mitochondria, on scotomorphogenesis in Arabidopsis. So I'm working with Livia Merendino and Bartro Gakiar, and we were working IPS2, as Pierre said earlier. So some of you might know about this scotomorphogenesis. So let me just remind you. So when the seeds are under the soil, just after the germination, they come through the soil. We know that. So this is the germination stage. But before coming out of the soil, and which is called the photomorphogenesis, there is another developmental stage under the soil or in the dark that is called the scotomorphogenesis. So let me say it again. So scotomorphogenesis is the development of the seedlings in the dark, just like under the soil. So when the seedlings are developing under the soil, they show this long hypocotyl in search of light, a non-green cotyledon due to lack of functional chloroplast and chlorophyll, and an apical hook here to protect the seedlings with the friction from the soil. So this apical hook is very important for the seedlings to come out of the soil and survive after the emergence. So our lab, my team, they have identified before my arrival that mitochondria play a very important role in this scotomorphogenesis control. So that means that we have used the mitochondrial respiration mutant, and what we have found that when there is a problem in the mitochondria, there is significant alteration in the developmental response. So you can see here that there is this alteration of the apical hook, and we call it as the twisted phenotype. And the hypocotyl, they are very small, and also the roots, they're very small. and we call it as developmental response or the twisting phenotype. So if we go further, what's happening inside the cell, what we identified that when there is the mutant of the mitochondrial respiration, when the electron transport chain is not working or functional, then the mitochondria sends the signal to the nucleus through a transcription factor called the NAC017. And then you have the alteration of NAC017 mediated mitochondrial stress markers and also the alternative oxidase or AOX. So these two are very important for this developmental response, and in a sentence, the mitochondria play a very important role in scotomorphogenesis. Now, what we know that in addition to mitochondria, there is another bioenergetic organelle in the cell, that is plastid. And the plastid in the dark is called the ethioplast. And there are many similarities between the plastid and mitochondria. For example, they have their own genome, and they also share different polymerases. And in addition to that, they can produce the energy. We all know about the mitochondria because they can produce the energy through the mitorespiration. But the ethioplast, which is the plastid in the dark, can also produce the energy through the ethiorespiration. In addition to that, they are connected with each other by the different metabolites, and they are connected through the nucleus through the pathway called retrograde and anterograde pathway. So since our team already found that there is the role of mitochondria in this scotomorphogenesis, that's why we will try to understand what might be the role of the plastid or the ethioplast in this scotomorphogenesis. So the idea was that if we block the plastid gene expression, then what happens? And the second one is that how the plastid can be connected with the nucleus. What are the signaling factors that connects from the plastid to the nucleus? So to answer the first question, so the strategy we followed is here. So if we look into the genome structure of the plastid or the ethioplast, we can see that there are two different types of polymerase. So this is the NAP, which is the nuclear encoded polymerase, and this is PEP, which is the plastidial encoded polymerase. And we can block the translation of both these type of polymerase by the antibiotics called the lincomycin and the spectinomycin. So using this lincomycin and spectinomycin, we are using, and we are trying to find out that what might be the developmental response and what might be the signaling factors that are connecting the plastid to the nucleus. So the first result comes here. If you see here that this is the wild type control, which show a normal scotomorphogenesis phenotype, but when we are blocking the plastid gene expression with the lincomycin or the spectinomycin, we have the significant alteration here. So that means a twisting phenotype here at the apical hook angle. So then we quantified this apical hook angle here, and then we found that the lincomycin and spectinomycin is increasing or altering the apical hook angle. And so you might be thinking that if the lincomycin and spectinomycin is really affecting the plastidial genes or not. So what we did the plot to find out that yes, the lincomycin and spectinomycin, they're blocking the RBCL, that rubiscolar subunit. And we could also see another thing, that when we are treating with the lincomycin or spectinomycin, the fluorescence level of protochlorophyllide is significantly less. That means the protochlorophyllide is the precursor of the chlorophyll. So which means that we are blocking the plastid gene expression, and it's really working, and it's working on the chloroplast at different developmental stages. So then we went for the structural point of view, because we know that the ethioplast has a very interesting structure with the PLB. So this is the prolamellar body and the prothylakoid, and this prothylakoid becomes thylakoid in the light. So we wanted to see the PLB structure and the prothylakoid, and we found that when we are treating with the lincomycin and spectinomycin, we could see that there is the PLB structure. You can see here in the left, this is the control with a very nice PLB structure. But when we are treating with the lincomycin or the spectinomycin, the PLB structure is completely disrupted. And we see a sponge-like PLB structure here. And also the PLB is very small, and we have a lot of starch grain here. So then we wanted to see at the gene expression level what's happening. So we found again that the genes which are related to the plastid, they are downregulated. So it's normal that we are using the plastid specific antibiotics, and we could see that the downregulation of plastidial genes. And interestingly, so among those two antibiotics, the spectinomycin and lincomycin, we could see that there is some difference between their mode of action. The lincomycin is really the plastid specific. It's not affecting any other organelles like mitochondria. So here you can see that there are some of the genes related to mitochondria, which are mitochondrial encoded genes. They are upregulated in the spectinomycin treated seedlings, but not in the lincomycin treated ones. So it means that maybe there might be some other effect of the spectinomycin, but not the lincomycin. So we wanted to look further what's happening and compare between those antibiotics. So yes, so then we wanted to see that, okay, since lincomycin is affecting mitochondria, lincomycin is not affecting, but spectinomycin is affecting mitochondria is little bit. So if the redox status or the metabolism is affected or not. So we used a very nice tool that is the biosensor line of the mitochondrial ROS. So these are the lines. They will give you the signal when there is the significant alteration or the alteration of the mitochondrial redox system. So what we could see with that with the lincomycin, we didn't see any difference of those alteration of ROS amount, but with the spectinomycin, there was significant increase in the ROS amount. So that means and this is the mitochondria one. So that means the spectinomycin might affect or is affecting the mitochondrial redox metabolism. Then we went for the GCMS analysis. Interestingly, so we did the GCMS with the lincomycin and spectinomycin treated seedlings, and what we found that with the spectinomycin, the amount of or the accumulation of TCM metabolites is significantly increased rather than the lincomycin. So this was another assurance for us that yes, the lincomycin is affecting only the plastidial one, and on the other hand, the spectinomycin is affecting both the plastid and the mitochondrial metabolites and also the redox system. We also quantified the level of carbohydrates and the fatty acid with the GCMS, and we found that the same type of result, that is spectinomycin, but not lincomycin, results in the accumulation of the sugars and the fatty acids. So that means that the mitochondria is getting affected by the spectinomycin, and lincomycin is affecting only the plastid. So we wanted to see, and we know that if there is any problem in the mitochondria or the plastid, the nucleus responds. So we wanted to check how the nucleus is responding here. So then we checked the genes related to photosynthesis, which are encoded in the nucleus, and we found that in case of both the antibiotics, the genes related to photosynthesis are down. And then we checked the genes related to mitochondrial stress. So we found that when we are treating with the spectinomycin, the genes related to the mitochondrial stress is significantly up, but not for the lincomycin. And this effect is same as just like another mutant of mitochondria. That means we have another mutant of mitochondria who is showing the same type of response like the spectinomycin, but not the lincomycin one. So that means we were quite sure that okay, we are getting we are blocking the plastid gene expression and with the mitochondria a little bit probably, and so we are also getting the nuclear response. So then we wanted to look at the signals, the retrograde and the anterograde signals. So we know that from the ethioplast or the plastid, if we have a problem in the plastid, then the retrograde is mainly the GAN1 mediated, and if the mitochondria is being affected, then the retrograde is mainly the NR017 transcription factor mediated. So then we checked and treated the GAN1 mutant and the NR017 mutant with the same antibiotics, the lincomycin and spectinomycin. And here you can see here that the lincomycin and spectinomycin, they really don't affect the twisting phenotype. And for the NR017, you can see that the spectinomycin is affecting the mutant of NR017. So which indicates, I mean, let me be refine you, tell you again that when you don't have the GAN1, that means when you don't have the connection between the plastid and the nucleus, then the seedlings are not showing any twisting. And we could see also at the gene expression level that with the lincomycin, there is no alteration of the photosynthesis associated genes, but for the spectinomycin, there is still some down regulation. And for the lincomycin, we could still not found any difference between the wild type and the NAC mutant, but for the spectinomycin, we could see that we still have some response at the nucleus level. So what we think right now that probably when we are using the spectinomycin, it might be a direct effect or the indirect effect. It can go directly to the mitochondria, or it might be affect, or it might affect only the plastid and then go through the nucleus to the mitochondria. So that's why we're trying to find or utilize the double mutant of GAN1 and NR017 right now. But let me summarize what I told you, that when the scotomorph, this is the in the regular scotomorphogenesis, when you have a normal phenotype with the apical hook, and if you block the plastid gene expression with either lincomycin or the spectinomycin, you have the alteration of the nuclear genes, and for both of them, the GAN1 is mediating this alteration. And in addition to that, for the spectinomycin, we could see the alteration of the mitochondrial metabolism and mitochondrial ROS, and this is probably being regulated by the NR017. But we still need to find out the signaling, if it is both GAN1 or the NR for the spectinomycin or not. So I would like to thank you for your kind attention, and thank my supervisors, Livia and Bartro, our team, and our collaborators, Lucia and Mihao from Warsaw University, and Marcus Schwarzlander and Pedro Barreto from Münster University, Germany, with whom I worked for the redox metabolism and also the structural point of view. And I thank you for your kind attention.
Thank you, Sadek. Perfect timing. Any questions? I do have one.
Yeah.
So basically, you conducted targeted metabolomics on specific compounds like sugars, well, primary compounds. I wonder if you also did targeted approaches to get like signals, maybe regulators in the context of your study.
No, we didn't do that. So it was mainly the primary metabolites on which we focused. But I think we have this point, and we will work on this, especially the untargeted metabolomics, very soon. Yeah.
And what about the redox compounds?
The redox, it was mainly the biosensors lines which we used, and in addition, we are also quantifying the NAD level, NADAs, and the other pyridine nucleotides level like the NADP and NADPase, and we are also quantifying the antioxidants but yeah, so we are in the process of quantifying them.
Okay, thank you.
So thanks a lot. So now we have our last speaker. So Carolina Dalmeida is a PhD student at UNARIO, Rio de Janeiro, and research associate at the Center of Innovation in Mass Spectrometry. So I think you can share your screen now, and the floor is yours.
Can you see my presentation?
We can see your screen for now.
Oh,
So that's your conference, but no, the presentation.
sorry, I'll try to change. Okay.
Yes.
Just a moment. Okay. So thank you for the presentation, for the introduction, and thank you for the invitation to be here today.
Just it's not in full screen, but I don't know if it's
no.
Yeah, no, it's not in full screen.
Oh. I will show just a moment. I will try a different way to share. Okay, it's working?
Yes, perfect, thanks.
Okay. So thank you for the introduction and for the invitation to be here today. I'm Carolina Dalmeida. I'm a PhD student at Unirio, Brazil. And today I will talk a little about the bioactive potential of sorghum grains for human consumption by metabolomic approach. Just a briefly introduction, a briefly context. The world faces some challenge in food supply. We have an increase in non-communicable disease, mainly caused by unhealthy diets. And we also have an increase in temperature, the global temperature. On the other hand, we have a stagnation of the major crops such as wheat, rice, and maize. But we also have an increase in global population estimated for the end of this century. Based on this, food science has a dilemma: how to feed a growing population with healthy food sustainably. The use of non-conventional cereals such as sorghum grains can be a solution for this. Sorghum has some interesting aspects such as agronomic and nutritional aspects, but today I would like to highlight the health aspects, mainly caused by biological compounds, mainly phenolic compounds. Sorghum grains has higher phenolic compounds than conventional cereals such as wheat, and that stimulates the use of sorghum for human nutrition. And today we already have several sorghum-based products in the market. But some gaps about sorghum phenolics needs to be need to be filled. So the first one is, how does the phenolic compounds change through the sorghum grains maturation? And thinking about the final products, what impacts does technological processing have on the phenolic compounds of major sorghum grains? To answer this question, sensitivity and exploratory analysis need to be applied. For example, the use of on-target omic tools using data-independent acquisition, DIA, can be a good strategy to elucidate the synthesis of these phenolic compounds during the grain maturation and to understand the technological impacts on its phenolics profile. So 3 methodological steps were applied in the present study. The first step is the extraction, the phenolic compounds extraction. So these phenolic compounds are in the outer layer of the sorghum grains, the pericarp, and they can be found in free and bound forms. So based on this, we perform a sequential extraction. First, for the extraction of free compounds, we used 80% ethanol, and after we apply hydrolysis to obtain the bound phenolics. These extracts follow to the UHPLC system, coupled to a mass spectrometer, Q-TOF type, with a ionization source of electrospray in negative mode. And between the two analyzers, we have a collision cell where we can apply a low and high energy of collision to annotate, to detect, and probably identify the precursor and fragment ions. After the third and last, the final step is the data processing. So we have a huge amount of data, and we have to process this information. So alignment, normalization, and annotation were performed using Waters Company Progeny software. In our lab, we do the DDA acquisition mode, and we also use OpenASSEC software, but in this study we use just this paid software. For annotation, we use a custom phenolic database by PubChem and Phenol Explorer. And our annotation was performed according to the Metabolomic standard initiative levels of identifications, but only features present in the 3 replicates and with the CV higher than 30% were annotated and quantified. Also, as info, XL stat, and meta-analysis were applied to multivariate statistical analysis. So thinking about the first question, how does the phenolic profile change through the sorghum grains maturation? We analyzed 5 different maturation states of sorghum grains, from milk to mature grains. So we have grains from 10 days after flowering to 40 days after flowering, to test the hypothesis of the variability of sorghum phenolic compounds during its grain development. Also, we analyzed 2 different genotypes, tannin-rich and tannin-poor, and 2 different growing seasons to test the influence of genotype and growing seasons on phenolic compounds variability. First, we tentatively identified 97 phenolic compounds. Here we can see a Venn diagram where the samples are plots, and we can see a difference, a huge difference between the genotype, the contrasting genotypes, but we can see a good separation, a good variability between growing seasons. When we analyze the maturation states during the grain growth, we can plot our totally identified compounds in a heat map where, based on ionic abundance, hierarchical clusters formed 3 large groups. The first one is the group where the phenolic compounds are synthesized in the beginning of maturation. Here we have mainly phenolic acids. The second group, we have the intermediate phenolic compounds. Here we have a similar proportion, so we have 1:1 phenolic acids and flavonoids. And the third group, we have the phenolic synthesized in the final stage of maturations, where we have 60% of flavonoids. Based on this, we can propose a hypothesis. So here we have the phenylpropanoid biosynthesis pathway, when we can say that In initial states, the conversion of phenylalanine and tyrosine, the phenolic compounds precursors, it's mainly to form phenolic acids. So we have the formation of phenolic acid by these two amino acids. But from the 25 days after flowering, we have the flavonoid biosynthesis pathway seems to be prioritized. So we didn't measure the enzymatic activity in our study, but probably in maturation, we have a greater action of enzyme 4-coumarat-coagulase. Well, after understanding how these phenolic compounds are synthesized during the grain growth, we begin to think about the second question. What impacts does technological processing have on the phenolic compounds of mature sorghum grains? To answer this question, we apply three different processings. We have germination as biological processing, we have derulling as physical processing, and we have cooking as thermal processing. To test this, the hypothesis of the change in phenolic compounds, maybe a change in solubility or levels after the three different technological processing. In biological processing, the first one, we can see here flavonoids as the majority class in our sample. And we also know that in mature grains, it's normal, it's what happens. But after a short germination, 72 hours, and a long germination, 144 hours, we can see a reduction in flavonoids contents. So in the final stage of germination, we have a similar proportion of a similar ratio between flavonoids and phenolic acids. But an interesting find in the study is the presence of procyanidin, also known as condensate tannin, in the sample genetically classified as tannin-free. So here we have Macia, a sorghum genotype well known as tannin-free genotype, and we detect some procyanidins even in this genotype. In the genotype rich in tannins, we can see a decrease in procyanidins contents after germination. These finds are important because procyanidins can complex with proteins and starch of sorghum grains and reduce their digestibility. So for human nutrition, it's crucial to understand these phenolic compounds. During the thermal process, we analyzed 4 different genotypes with different pericarp colors, and we didn't saw a variation in the phenolic profile after 30 minutes. So thermal processing, it's a processing that didn't change so much the phenolic profile. Finally, the last processing was the dehulling. First of all, our sorghum sample has like a husk in the composition, the outer layer. So we remove this husk manually, and we analyze this fraction of the grain. So here in the husk, we have plant pigments present in this layer of the grain. And after the hulling, we can see that we reduce our phenolic acids of the sample, important phenolic acid as caffeic acid and ferulic acid. It's not so good from a biological point of view, but when we analyze the flavonoids, we saw a loss of 9 isomers of procyanidins between dimers and trimers. And as I told, procyanidins are related to a bad starch and protein digestibility. So even if the ruled grain has less bioactivity compounds, it can be associated with a higher digestibility. It's very important for human nutrition. Just one final message. The phenolic stud for the grain maturation was elucidated for the first time in sorghum grains, and using on-target metabolomic, we can see the difference between the balanced phenolic acids and flavonoids during the grain growth. Analyzing the final products, the final sorghum-based products, we can see that we have similarities and difference between the grain processing, but they had in common the decrease in flavonoids contents. We believe that our work can stimulate new sorghum-based products with the better selection of the processing technique applied, and maybe the use of immature grains in sorghum-based markets. I thank my supervisors, Mariana Ferreira and Amzama Mehim, and also all the collaborators. I would like to answer also the financial support, and mainly RFMF for the opportunity to be here today, and also for the financial support in the last year, 2023, where I went to Global Sorghum Conference in Montpellier, France. I presented there 2 oral presentations, and I won the first place in 3-minute stats competition. So really thank you, and I'm open to questions.
Thanks a lot for your presentation. So do you have questions? No, so maybe I have one little. So on your analysis, you use negative mode for ionization. So why do you use negative mode? Why did you choose that?
Previously in our lab, we tested our extracts with positive and negative ionization modes, and we have better results with negative modes. So it's a protocol of our lab. In our lab, we work just with phenolic compounds, the focals of the lab. So it's like a methodological protocol of our lab based on previously results.
Okay. Thanks. So I don't know if there is other question. No. So thanks for all the speakers for this webinar. So thanks a lot. The next webinar will be in September. So we will email the different information of the next webinar soon. So thanks everyone, and thanks for the whole speaker. And see you in September.
Have a good summer. Bye.
Thank you.
Thank you for you too.
Thank you. Bye.
Thank you. Bye.